Welcome to the MOOC's NPTEL course on mass spectrometry-based proteomics. Proteome describes the protein complement expressed by a genome. The extent of diversity and complexity due to alternative splicing and post-translation modification is tremendous. Therefore, studying proteins and proteome becomes very important. In recent years, mass spectrometry has played a major role in proteomics level investigation, and in 2014, two human proteome reference maps were published using high-resolution mass spectrometry. This course is divided into 4 modules. Each module will be finishing in 1 week. There will be 20 lectures of around 30 minutes duration, which will cover the key concepts, experiments, and laboratory demonstrations to explain the concept effectively to the students. The first module will discuss about the basic concepts of proteomics and the importance of sample preparation. Further, we'll discuss the fundamentals of mass spectrometry and the advancements in technology. Lecture 1 will focus on basic understanding of proteomics, the technological advancements in analytical techniques with increased sensitivity, resolution, and capability to carry out high throughput studies have led to the transition from protein chemistry to the new field of proteomics. How limitations of mass spectrometry for protein analysis was overcome by development of soft ionization techniques such as MALDI and ESI will be discussed in this lecture. In lecture 2, we'll talk about sample preparations for proteomics applications. Protein extraction is a crucial step for any proteomic investigation. The protein extraction method involves cell lysis, prevention of proteolysis during lysis, different type of protein precipitation methods, protein solubilization, and removal of various interfering components. The protocol standardization becomes a major challenge in proteomics, and there is no generic protocol which exists in literature which can be used for every sample type. Therefore, we'll discuss few sample preparation strategies and how a good sample can be prepared for the proteomic analysis. We'll continue the protocol standardization using bacteria and discuss about cell lysis, protein precipitation and quantification, and discuss about trisol extraction protocol. In lecture 4, we'll talk about in-gel digestion or in-solution digestion of the protein, which is essential prior to the mass spectrometry analysis. Often the in-gel digestion is used to extract proteins or peptides separated on the gel electrophoresis. In-gel proteolytic digestion, the in-gel proteolytic digestion is performed to cleave the protein of interest present within the polyacrylamide matrix. Mass spectrometric identification of the target protein greatly depends on the efficacy of in-gel digestion protocol, which generates mixture of peptides from the target protein through proteolytic digestion. In lecture 5, we'll talk about the fundamentals of mass spectrometry. Mass spec is highly sensitive, balanced to measure the mass of the molecules in vacuum based on mass to charge ratio. It consists of an ionization source to ionize the molecule a mass analyzer to resolve the ionized molecules in vacuum, and detector to read the signal coming from the mass analyzers. There are different ionization sources and mass analyzers being used for the proteomic analysis. If you know the principle of each one of these, it becomes much easier to select which configuration to use and when for your proteomic investigations. In second week, we'll discuss in detail about different type of chromatographic techniques like gel filtration, ion exchange chromatography, affinity chromatography, SCX or strong cation exchange, and reverse phase chromatography. As we will progress through lectures 6 and 7, we'll discuss how different chromatography techniques work based on different principles and how one could employ those in proteomics workflow. Lecture 8 will focus on ionization sources. The ionization sources are responsible for converting the analyte molecule into gas phase ions in vacuum. The ions generated by the ionization source are then integrated with the mass analyzer. The commonly used ionization sources are MALDI and ESI. In lecture 9, we'll talk about mass analyzers. The mass analyzers resolve the ions produced by ionization source on the basis of their mass to charge ratios. Various characteristics such as resolving power, accuracy, mass range, and speed determine the efficiency of these analyzers. The commonly used mass analyzers are time of flight, TOF, quadruples, and ion traps. The sample preparation strategy prior to mass spec analysis will be discussed in lecture 10. Once the protein sample has been digested, all the salt, buffers, and any detergent must be removed from the sample, which can be effectively performed by using some filters such as zip tips. It offers several advantages such as quick purification, sample enrichment, and ensures that there is no contamination. However, it can purify only a limited volume of the sample and also absorbs some amount of protein sample, thereby leading to losses. Further, we'll talk about how to use MALDI to analyze your samples in high throughput manner. The third module will cover Introduction to Quantitative Proteomics and Hybrid Mass Spec Configurations. The complexity and dynamic nature of proteome presents major technological challenges. Mass spectrometry advancements have improved the high throughput identification and quantification of proteins and now offer an opportunity to understand the human diseases and discover biomarkers. The lectures 11 and 12 will cover hybrid MS-MS configurations as well as discussion on two latest hybrid MS technologies, QTOF and Orbitrap. The basics of quantitative proteomic analysis and what are the different types of quantitation methods exist in literature using mass spectrometry will be discussed. In lecture 13, we'll talk about quantitative proteomic analysis using stable isotope labeling by amino acids in cell culture or SILAC. SILAC is an in vivo labeling method. The labels can be introduced in vivo by growing an organism in a media enriched with specific isotopes. There are different ways of in vivo labeling, such as enrichment of 15 nitrogen media, culture-derived isotope tags, known as CDIT or SciLac. In this lecture, the major emphasis will be on SciLac method for quantitative proteomic analysis. The next lecture will talk about quantitative proteomic using ITRAC technique. ITRAC involves identification and quantification of complex protein mixtures by MS-based quantitative proteomic techniques. The ITRAC reagent consists of amine-specific stable isotope reagent which can label peptides of up to 4 or different biological samples. The ITRAC method provides multiplexing capability of 4 or 8 sample analysis, which is not possible using ICAT, where only 2 samples can be labeled and analyzed. In ITRAC, 4-plex reagent set, the M-diz value ranging from 114 to 117. There is a balance group of mass 28 to 31 Dalton. Therefore, the overall mass of reporter and balancer component remains constant. During the MS-MS fragmentation, the reporter ions give the peaks at 114, 15, 16 and 17, which provides the information about the peptide or protein abundance. The next lecture will focus on another quantitative proteomic technique based on tandem mass stats known as TMT. TMT is also in vitro labeling method which is similar to ITRAC method. TMT is MS-MS based quantitative technique which uses the isotope labeled model referred as tandem mass stats. It provides the accurate quantification of peptides and proteins. By using different type of TMT tags, one could perform multiplexing experiments of 2, 4, 6 or 10-plex. In the last module, we'll talk about quantitative proteomic data analysis and some aspects of systems biology applications. The quantitative proteomic technologies aim to identify the differentially expressed proteins in a biological sample. The differential expression of proteins can be caused by a disease state or various external factors like stress, drugs, or different experimental conditions. The data analysis is an important step for protein identification and quantification in proteomics workflow. The accurate quantification of protein abundance becomes very important for the quantitative proteomic analysis. This lecture will focus on data analysis for the protein identification using mascot and protein quantification using ITRAC based workflow. The next 2 lectures, 17 and 18, will cover proteomics and systems biology. So what is systems biology? Systems biology is the examination of a biological entity as an integrated system rather than studying its individual characteristics, reaction and component, and that is what is termed as systems biology. The distinct approaches of systems biology include the model-based and data-based methods. The model-based approach involves some prior information which can be implemented in these models, whereas the data-based methodology, the objective is to find the new phenomenon. Some of these details will be covered in these lectures on systems biology. Proteomics has wide range of applications from understanding the physiology of microorganisms to biomarker discovery for cancer and other diseases. Towards the end, I would like to cover proteomics applications and challenges. The last lecture will focus on proteomic challenges. Mass spectrometry is one of the best inventions in proteomics field in recent years. It is able to achieve many milestones, including the draft human proteome maps. Though mass spec is a sophisticated instrument and provides very high throughput, robust capability of analyzing proteome, still it has many challenges to overcome in the future. The post-translational modification analysis using mass spec is one of the challenges. Additionally, the dynamic range of proteins, inadequate coverage of whole proteome, and accuracy of quantification are challenging in this field. Overall, this course will provide the basic knowledge of mass spectrometry with focus on quantitative proteomics. Hope these concepts and understanding will be useful for your research. Thank you.